All right, thank you everyone for joining. We have a nice talk for you today. We'll be, I'm introducing you to Dan Trieber. He's our Chief Technology Officer at DiscoverX, and he'll be speaking about creating cellular target engagement assays, and this is for diverse kinases using our new InCELL Pulse technology. Today's webinar is being recorded, and your phone lines have been muted. So if you have any questions, during this webinar, please use the Q&A area or the chat box within the WebEx Event Center application. As a reminder, the slides and the webinar recording will be emailed to you as early as tomorrow. So with that, I'd like to introduce to you Dr. Trieber. You ready, Dr. Trieber?
Yes, thank you very much, Dana, and thank you everybody for attending this webinar where we want to tell you a little bit about a new technology that we've developed called InCELL Pulse for looking at cellular target engagement for small molecules. And we've really focused on diverse kinases with this technology, and that's what I'm going to tell you about today. So the agenda for this talk, I'll first start off with a brief overview about the platform and the importance of cellular target engagement. And then I'm going to move into a kinase-focused section of the talk where I talk about a little bit about kinase drug discovery opportunities and some validation data for some diverse kinases that we've looked at with this platform. And then the question is, how do you access this technology? So I'll summarize some of the tools that we have available and the workflows for using these tools. So what is cellular target engagement? So cellular target engagement is a fundamental pharmacological principle. Does my compound enter the cell? Does it engage the protein target? And how potent is this interaction? It's a very fundamental question, yet it's often difficult to measure if there is not available a functional assay using a proximal biomarker type readout, for example, autophos for a receptor tyrosine kinase. So this can actually be very difficult to measure, and new technologies are needed to address this question during drug discovery campaigns, during target validation, chemical probe development and validation, etc. So what's our take on this? What have we done? So we've developed a platform we call InCELL Pulse. It's a thermodynamic-based assay that takes advantage of the fact that the thermal unfolding of a protein target is altered by the presence of a bound ligand. So if we focus on the right-hand side of this slide, we can envision here a thermal shift assay where we're increasing the temperature and watching the target protein inside a cell denature and aggregate as a function of the temperature. Now, in the presence of a ligand bound to that protein target, the TM for that transition shifts to a higher temperature. And what that allows us to do then is create an assay window where at particular temperatures, there should be a dose-dependent increase in the amount of folded, unaggregated protein as a function of that ligand concentration. And then shown on the right would be an isothermal example where we've chosen a temperature, say 50 degrees, and titrated in ligands that bind to the protein target of interest, and as a function of that ligand concentration, the amount of soluble protein is increased. So the real magic of how we do this is how we read these assays out. So it's not straightforward to read out the fraction of soluble protein inside of a cell. So we've applied a DiscoverX technology called enzyme fragment complementation to address this complex issue. Enzyme fragment complementation is based on a split beta-galactosidase system. So you could take beta-gal and break it into two pieces. One very small piece, a 40 amino acid peptide called prolabel, and then a very big piece called the enzyme acceptor. In and of themselves, these fragments have no enzyme activity, and with the appropriate substrates will not give a chemoluminescent signal. However, when they're combined, they complement and give signal. Now you can attach this pro-label tag, this small peptide, to a protein target of interest, and if that protein is denatured and aggregated, that complementation will be less efficient. So we can monitor the state of the protein based on the amount of complementation that's given by the enzyme acceptor addition. And so this is an engineered thermal stabilization assay using this DiscoverX technology. It's a very simple method. It's a mix and read luminescent readout method. And what I really like about it is that it's generic. It does not require any target-specific reagents like tracer molecules or antibodies or mass spec. It's very generic, and I think it has a nice place in this space. Moving on to the next slide, give a little bit more detail on how this works. So here's the cell. Again, this is an engineered system. The cell is expressing the target protein of interest, say a kinase, with that pro-label tag appended to it. Compound is added to the cells. The cells are put through a heat denaturation step, and in the absence of compound binding, that protein will denature, and that pro-label tag gets buried in this big aggregate, and there's poor complementation with the enzyme acceptor piece, and there's limited amount of luminescence. However, if the compound binds and shifts out that TM for aggregation for the target protein, there will be rescued complementation and will get light. So that the pro-label tag is really a very well suited for this application because it's a readout of the structural state of the target protein, and it's sensitive. Its detection is sensitive to aggregation. So what are some of the applications of this technology? So what I really like is that this technology can be applied across multiple phases of drug discovery. Certainly, it can be used to verify that compounds are cell permeable and engage the target. And that's very important when you're developing a probe molecule. When you're validating a target, one of the pillars of target validation is that you can show that in the relevant cell type, or at least in a generic cell type, that the compound gets into cells and interacts at some affinity with that target. It can also be used to rank order compounds during a lead optimization campaign. So, for example, in kinases, there are not functional cell-based assays for a significant fraction of the kinome. So looking at a direct thermodynamic measure of target engagement can be a very, very important assay to have in your back pocket. Now, we validated this technology across some diverse protein target classes, hydrolases, methyltransferases, and really focused on diverse kinases. And I'll get into why we focused on kinases in a minute here, actually right now. So I'm going to go through some validation data for the diverse kinases that we've looked at. And on the right side of this title slide is a dendrogram of the human kinome. Of course, there's 500 plus kinases. What we did was we obviously weren't going to build assays right away for 500 of these things. So we tried to build a diverse set of assays, and this is showing the little red globes show assays that we've built across this phylogenetic space. And so we tried to be very diverse. We did not just pick the usual suspect kinases, because my hope is that we can go beyond that, beyond tyrosine kinases. So why did we focus on kinases? So kinase drug discovery is a growing opportunity, and there have been 30 plus marketed inhibitors approved. It's the most approvals by the FDA of any target class in the past 10 years. But with that said, the field, in my opinion, has been a little stagnated. And what's been holding that back? Why are we just focusing on the same usual suspects, the same 15 kinases? Well, one thing is that we needed some new application for kinase inhibitors. And one of these new applications that we're all very excited about is ion. And so there were data showing that checkpoint inhibitors, which are biologics, combined with kinase inhibitors can have an additive, maybe even synergistic effect, and there's some really complementary biology there. Secondly, folks at the Structural Genomics Consortium, the SGC, who we collaborate with quite closely, have been asking the question, why is everyone only studying 15% of kinases when there's another 85% out there? And what they identified was that the limiting step was a lack of potent and selective tool compounds, chemical probes, for the understudied kinome. And so they've started an initiative that's already very successful, where they're creating incredibly valuable chemogenomic sets to ask the question, what do the rest of the kinases do, and what diseases are they relevant for? And of course, to support this initiative, there's a real need for kinase cellular target engagement assays, so that when they develop a probe, it can be validated in the cellular milieu. And I'll show some data on how InCELL Pulse has helped move that initiative forward. So InCELL Pulse for kinase drug discovery. Like I said, there's a need for cell-based kinase assays, generic assays, and so we decided to apply InCELL Pulse to this problem. We have a lot of kinase expertise. Of course, my group developed KinomeScan, which is a biochemical platform. We have tons of inhibitor data that we could leverage across this initiative. And when we decided to create this validation panel, I wanted to look broadly. I wanted to include some well-studied kinases, for example, ABL, and that's just to kick the tires. That's just to show folks that this technology is validated. Everyone understands the ABL data in the literature, and so that will provide some very nice proof of concept. I also wanted to include some understudied kinases that might have some good applications to new disease indications in IONC. And then the other question I get all the time at KinomeScan is, hey, you know, most of the compounds that we test hit these 3 or 4 kinases I've never heard of. You know, does this matter? What are the ramifications of this in cells? And the first question you want to ask is, well, does the compound actually engage the target in cells? So I included some common off-target kinases here. For example, GAK, G-A-K. That kinase is hit by almost 50% of kinase inhibitors that we look at, and so that's an important one for de-risking to look at, and also to understand maybe there's some disease applications for that as well. So I'm going to go through briefly some kinase assay validation data that we've collected using InCELL Pulse, and just to show that the technology is robust and works across diverse kinases and inhibitor types. I'm only going to focus on 4 or 5 examples here, but to the right, I show an image of a document that we've put together called the Kinase Reference Set for InCELL Pulse that has validation data for all 25 kinases on the list I'm showing here on the right-hand side of this slide. So this document is extremely valuable. It can be downloaded at the URL shown below, and the validated expression vectors for these kinases are available. So I'm going to focus first on monitoring or measuring data for diverse inhibitor types. So for ABL and BTK and MEK, we've got examples of non-ATP competitive type 1s, type 2s, and irreversibles. And then FAC is a kinase that really intrigues me. I was talking about how IONC is now a really significant area of interest for kinases, and FAC is actually connected to IONC with some pretty compelling data. And FAC is also a common off-target when we do biochemical inhibitor screens on diverse inhibitors. So there's a lot of opportunities here. And then I want to focus on some understudied kinases where we're working with the SGC to help them build potent and selective probe compounds that have cellular activity, and I'll show you a little bit of case study data on this. So for ABL, you know, just this is a kick the tires type approach. Can we measure type 1 and type 2 inhibitors? Can we measure relevant potencies for these inhibitors? And of course, the answer is yes. Dasatinib, extremely potent type 1 inhibitor in cells, sub 10 nanomolar EC50. Imatinib, of course, one of the first FDA-approved kinase inhibitors, type 2 inhibitor, a little less potent, agreeing with the literature. Negative control lipid kinase inhibitor, PI103, shows no activity. What was really exciting was that we can also detect binding of a allosteric ABL inhibitor, GNF2, that binds at the myristate site, and there's been some recent excitement about this class of inhibitors in the literature recently because the mutational spectra that would create resistance to these inhibitors is very different than from the active site binders. So combinations of these different paradigms could be very nice going forward. And so it was satisfying to see that this method can detect inhibitors outside of the active site. BTK and MEK, of course, obviously very important kinases. There's FDA-approved drugs for both of these targets. MEK1, the non-ATP competitive inhibitors are the ones that have been FDA approved, and because they do not compete with ATP, they're extremely potent in cell-based assays. They don't have to fend off the 1 millimolar ATP in a cell. CI-1040 was a first-generation non-ATP competitive inhibitor, and it's got reasonable, 100 nanomolar-ish cellular potency in our system, and then the FDA-approved drug trametinib, which is a second or third generation MEK inhibitor, is significantly more potent, 2.5 nanomolar cellular activity. For BTK, we looked at both a reversible and an irreversible inhibitor. Ibrutinib, of course, FDA approved, extremely potent in the cellular milieu, as might be expected because of its irreversible mechanism, 1 nanomolar-ish EC50. The reversible inhibitor being a little less potent, probably because it's reversible. Moving on to FAC, the Immuno-Oncology target. So there are data in the literature and on the posters I've seen, and companies have programs around this. FAC inhibition can combine very nicely with checkpoint inhibitors, with biologics targeting PD-1. So I think this is going to be a pretty hot target moving forward, and I'm sure there will be more kinases that fall into this class as well. So I decided to go after this one in an InCELL Pulse assay, and so we measured KDs for 2 very different compounds. One compound from Pfizer, the PF562271, was developed as a dedicated FAC inhibitor, and of course we measure very nice, sub 30 nanomolar potency for this compound. And then there's TAE684, which is described as an ALK inhibitor. However, when we screened this compound in a kinome scan, panel-wide screen, kinome-wide screen, we saw that this compound also had potent FAC activity, but that's just a biochemical result. It's really nice to then validate that there's some cellular activity, and it turns out this compound is just as potent against FAC in cells as the dedicated inhibitor. So I think this is an example of how you can take advantage of looking at some of maybe the off-target effects of your compound to jumpstart new programs, quickly move into a cellular assay to say, hey, this compound does engage the target in a cell, and this could be a very good opportunistic starting point for a new program, for example, an IONC. Moving on to the case study where we've worked with the SGC closely, not only to screen their libraries in our biochemical format, but also to validate that some of their probe molecules have cellular activity. So we've worked with them on 2 understudied kinases, GAC, which I've told you is a common off-target hit for multiple kinase inhibitors. So they have an initiative to build a potent and selective GAC probe, and they've done that biochemically. They have multiple compounds that are potent and selective in biochemical assays, but they cannot nominate a probe until they know that there's also potent cell-based activity or in target engagement. That's one of the pillars of probe validation. So we've looked at a number of their compounds and rank ordered the cellular potency. And as you can see, they've got potent and selective inhibitors that have very potent cell-based activity from 45 nanomolar, and then of course the SAR series goes all the way up to compounds that don't really bind at 10 micromolar. So these types of data have really enabled their efforts on nominating a GAC probe. They'll be, I think, publishing this data, and I acknowledge the folks I've worked with from the SGC, David, Caro, Bill, and Chris. It's been a wonderful collaboration. We've also worked a little bit with David on the HASPIN project, where they're in an earlier stage than they were with GAC. They've got some lead compounds based on biochemical data, and they just wanted to confirm that the different series had cellular activity as well. So that's a very nice evolving story. So how do you access InCELL Pulse technology? So I'm going to go through some of the products we offer and some different options for accessing the technology. The various different types of products we have are listed in this table. We do have a couple of target-specific assay-ready kits. These include frozen transfected cells that you plate the day before the experiment and you run the experiment the next day, like right out of the box. So we have assays for ABL and then for the MTH1 hydrolase. And these assays are really available if you want to evaluate the technology. I mean, you might be interested in these exact targets. We've already had some purchases just for that reason. But we also have them just so you can evaluate the technology and decide, am I going to go to the next step? And the next step would be for your own target, this do-it-yourself kit that we offer, which we call the InCELL Pulse Target Engagement Starter Kit. This is where you create your own assays. So I'll go into some details on this, but this is really the main product that we offer. And of course, we offer detection kits. Once you run out of detection reagents that come with this kit, we can resupply those once you start doing your screening. We also, for the kinases, so I showed that we have assays that are validated for 25 kinases. The validated expression vectors for these 25 kinases are available upon request as well. And these, combined with the do-it-yourself kit, will get you an assay for any of those kinases. And there's also the option of contacting us, and if you want us to build an assay for you, we can do custom assay development as well. So what is this starter kit, this do-it-yourself kit? What comes with this? So the parental vectors, so if we look at the table on the right, we provide the vectors for cloning your target protein of interest into this vector using the pro-label tag either on the N-terminus or C-terminus of the protein. We also include an MTH1 expression vector as a positive control while you're doing your experiments. It comes with detection reagents, enough for, I believe, 400 data points, and then some other control reagents and plates just to get you started. And so really, when things go well, you can get a good handle on developing your own InCELL Pulse assay in less than 2 weeks. After the cloning step, you can try your target as both a full-length construct or just a ligand binding domain only. I'll comment on that a little bit more in a minute. You could pick a custom cell type depending on what your target is and what you think the right background is. And also what's nice about this is you do not necessarily have to go all the way to stable cell line development. These assays can be run using transient transfections where you transfect on day 1 and read out the data on day 2 rather than going through and making a stable cell line, which can take a bit longer, but could pay dividends in some cases. So here's the workflow for creating your own assay using this kit. So there's the cloning steps and the transfections, of course. And because of the way this technology works, and it's based on thermal stabilization, we do recommend that you attempt both a full-length protein and, importantly, a domain-only construct. And the reason for that is that the ligand binding to the particular domain may not protect other domains in a full-length protein from thermal denaturation. So I think it's important there to think closely and carefully about what constructs you clone. Then there's some assay development steps that we guide you through. It's a very detailed user manual. So then there's those steps, and then there's the optional step at the end of making a stable cell line. So here's some scientific content I want to bring to your attention. So a team from GSK had a poster where they've incorporated InCELL Pulse technology into their screening workflow. So InCELL Pulse can be adapted to 384 well plates for screening applications. So that's that poster. There's links for all of these documents shown below. We've also had multiple posters, including 2017 SLAS. And then, as I mentioned earlier, we have this reference set for the 25 validated kinases that you can download as well. And these expression vectors for these assays are available, and we'll give you a jump start at building assays for any of these targets when combined with the do-it-yourself kit. So just to summarize, I've told you about this new technology, this generic cell-based target engagement technology called InCELL Pulse. We focused today on kinases. I showed the dendrogram again on the right, showing that this technology has worked across diverse kinases, across the phylogenetic tree. And it's a simple method, and again, what I really like about it is how generic it is. There's no requirement for target-specific chemical tracers, antibodies. There's no mass spec, and so I think it's really a convenient solution when a robust functional assay is not available. And with that, I will stop, and I'll be happy to address any questions.
Awesome. Thank you, Dan. We do have a few questions, time for about 2 or 3. One is one you kind of already answered. It's related to can you use higher throughput type of formats for InCELL Pulse other than 96 well?
Yes, so that's a great question that we get a lot. And yeah, the poster I show from the GSK folks, they've moved into those types of formats. So there are 384 well thermal cyclers for the denaturation step, and of course this is a mix and read type readout, so the plate can come right off the 384 well thermocycler, mix and read the detection reagents, and it goes right on a luminometer like an Envision.
One question, let's see, it says, can you comment on the different assay signal between kinase targets as well as a different assay window? Does that make sense? So between different targets, how is the assay window, like the signal to background, I'm assuming?
Oh, for different targets. Yeah.
Yeah, like how do they vary?
Yeah, so our standard for validating an assay is an assay window of at least 2 to 2.5. Most of them are greater than 3. Yeah, I mean it's like anything else. Some targets, some assays are going to give bigger windows than others, and that can depend on a number of different factors that are only incompletely understood. But yeah, the precision of the assays is such that even with windows of 1.5 or so, you can measure dose response curves.
Okay. This one's about the cells, like when they're heated, are they still viable? during, I guess, the PCR step?
Yeah, so we did a very, so we focused on HEC293 cells. These are actually pretty difficult studies to do to show detailed viability. So we focused just on one cell line. So we can go up to close to 60 degrees without suffering for 3 or 4 minutes without suffering significant loss in viability as measured by trypan blue dye exclusion. So I think, if I'm recalling, you could go 55 degrees for up to 4 minutes, but at 60 degrees, you really wanted to stay to 3 minutes. And other folks in the field using these types of assays have done this on more cell lines. But I would certainly recommend, if you're not using HEC cells, to evaluate this. In the user manual for the do-it-yourself kit, we talk about the data we collected for the HEC cells.
Okay, this one is about cell viability affecting how the compound interacts, I guess, with the cell. I'm assuming the target, but it says in the question, with the cell.
What's the question?
Cell viability, okay, it says, does the cell viability affect how the compound interacts with the cell?
Yeah, so we haven't really looked at that because we've only measured interactions under conditions where the cells are viable. Now, you could imagine cases, maybe you're looking at a membrane-bound target where there isn't cell penetration required. Then maybe you don't care if the cells are viable. You're almost just using them as membranes. But no, we haven't done a lot of work. All the data that I've shown are under conditions where the cells are still excluding trypan blue.
Okay, there's quite a few questions, so maybe we just have time for one more.
Okay.
Okay, how do InCELL Pulse kinase assays relate to the biochemical data?
Yeah, I get that one all the time. So it's pretty remarkable. So the compound set that we used for assay validation is a privileged compound set where we know the compounds have good drug-like activity, or good drug-like properties. They get into cells. And so for these compounds, we're seeing very good rank order potency agreement. We are seeing offsets between the biochemical and cellular, which is very typical for kinases, of course, because of, in particular, the ATP competition. So we're seeing 10 to 100-fold in general offsets between the biochemical and the cellular potency, with the biochemical, of course, more potent. Now, that isn't the case for the irreversible and the non-ATP competitive, where the biochemical and the cellular numbers we're measuring come out pretty much dead on, and that's exactly what you'd expect. So we do think that the offsets can be explained to some extent by competition with ATP in the cell. So good rank order, potency agreement, and offsets that you'd expect, and the right types of behavior for irreversible and non-ATP competitive inhibitors.
Okay, great. I do see other questions, which I will get back to. Dan and I will get back to you today. We have your email address, and I have the questions. So with that, I'd like to thank you all for joining today's talk on target engagement, and hope to hear or hope you join us on a future webinar. Thank you, everyone.